Allora, eccoci all'ultimo.
So this brings us to the last presentation before the debate. I had the opportunity to introduce myself already. My name is Alessandro Ricci, and I work with the members of this association, providing, let's say, scientific support to discuss the strategies, to take stock of new developments, with a view to being as, let's say, effective as we possibly can in disseminating information on PCDH19 so as to ensure that there is growing awareness with respect to what is, as we've learned today, a very complex condition. The aim, of course, is also to raise funds for further research work, which hopefully is going to be proof of concept for new therapeutic approaches. The purpose of my presentation, which is the result of a joint effort together with Francesca, is to look at things from a slightly different point of view. We've talked about many topics. We've seen that there are many studies that have, let's say, I think, shown us that there's a lot going on, and this is wonderful. Obviously, we have scientists and researchers who produce data, and then you have families and ordinary people who, let's say, become aware of the results of this scientific research. And depending on how this scientific research is disseminated, then expectations can differ. I would like to start by summarizing what Insieme per la Ricerca has done so far. It was founded in 2008, but between 2013 and 2018, the foundation has funded projects amounting to more than 400,000 euros, and this is exceptional, especially in Italy, where it is very hard to find funding. We have to often use international European funding. And we know that innovation and research are often terms only used by politicians seeking to be elected, but then there's very little by way of a practical impact. I joined this group 3 years ago. I saw how much had been achieved, and I thought that was amazing. I started reading the reports that were the outcome of these research projects, and I developed a growing interest with respect to this disease, and I also understood better how things were evolving. For the first time in 2018, we funded a project that for the first time had the term treatment in the title, and this was obviously a milestone of significance. The data of the meeting organized together is summarized here. We selected 6 authors for today from the most representative groups that have worked the longest and that are accredited in studying PCDH19. We received a request from Marinus Pharma, and Marinus is the only one to be conducting clinical trials. One has already been carried out. One is about to be launched. We received abstracts and selected 6 on other, say, PCDH19 studies, and we've decided to include these in tomorrow's session because we felt it was important for these research groups to have the opportunity to present their data. We also received an abstract, as was pointed out, on the link between epilepsy and the intestine, and we consider that this is an interesting research area. What is the state of play at the moment? What do we think we know? What have these studies indicated to, say, non-scientists? Well, the situation indicates that there are significant impacts during neuronal development and intracellular communication, and we know that this is at the heart of mosaicism that can arise. We've seen that there's a growing importance in synapse formation and maintenance, and this is something that has to be investigated. We've also seen that there are changes in dendrites and morphology. We've seen that there is a possibility of binding with the modulator of the GABA subunit alpha receptor. We know that there's a link with symmetrical and asymmetrical cell division during development. And we also know that it is a modulator of gene expression after protease cleavage, and therefore we have to identify the genes that are modulated. So in a manner of speaking, we're beginning to see that probably this protein, which is multifactorial and has many functions, is a critical core, let's say, element, and as Joseph showed us in two homozygotic twins, outcomes can be totally different. And Joseph showed this very effectively. This could be due with interactions with the chromosome X. As we've said, it is a random process in humans, but this can also be due to a number of other epigenetic factors. So, what we've begun to consider is that every patient is potentially unique. This is what studies on penetrance show us. What have we learnt today? What have we learnt this afternoon? Well, models are important to test new treatments. And finally, on PCDH19, we have seen that there's a lot going on, and there's some good stuff out there. We have stem cells with which we can carry out mixing experiments. We have cultures that are two-dimensional. We can therefore use these cultures to simulate mixing, wild type, and look at other possibilities. We have 3D organoids. Let's start by saying that some abstracts, and this has been something that has emerged for a while now, indicate that there is the possibility of using mouse models that can mimic quite well the disease. But as Jack showed us, in this particular context, there's a huge difference between humans and mice. So we have to examine these models very carefully, get to know them well. They have to also be very well characterized with respect to behavioral disorders and autism spectrum disorders. It's not enough to have a mouse model that can spontaneously induce seizures. However, this is encouraging, because in 2011 we only had a handful of models. Now we have a broad range of models that can be potentially useful to shed light on a number of issues and indicate what new treatment opportunities lie before us. What are the next steps? Well, ours is a small, not-for-profit organization. Well, it's not that small, but anyway, we have to promote a number of activities, and with the abstracts, having reviewed the literature, as we heard today, in fact, there are two possible paths ahead. There is no definitive, let's say, treatment available for patients at the moment, but there certainly is a potential pharmacological path that needs to be explored, possibly with drugs that have already been approved, but must be also used to treat PCDH19. So that would be an off-label use. But to understand what the right drug is, we need to better understand cellular, intracellular, and extracellular activities that can mediate and alter PCDH19. And one important point is how is the expression of other PCDH19 gene expressing PCDH expressing genes regulated. And another interesting possibility is gene therapy, which is going from lab to bedside. This is not any longer an academic issue. It is becoming more tangible. We wonder whether this is possible. In the light of today's presentations, the answer is yes. And we know that these types of research for both types of approaches takes a long time. We want to believe that this is viable. And to better optimize our therapeutic strategies, to have new drugs, we need more pure research to identify the affected genes, with which PCDH can interfere and which PCDH can modulate. To achieve this, obviously, we find that events such as today's are particularly important because we bring together world's leading experts on the topic of PCDH19. We also invite them to discuss amongst themselves and compare notes, as it were. We hope that there will be further research, that there will be proof of concept for new treatment approaches, and hopefully this can also attract pharmaceutical companies. And we're delighted, of course, that we have a representative from Marinas Pharmaceuticals that is carrying out a phase 3 study. And this means a possibility of using a biomarker to evaluate, let's say, clinical elements that can give us more information. The other path is gene therapy. We talked about antisense and other genetic technologies that we've been reading about in Nature only recently, and this was quite fascinating, of course, and we were very excited about what we were reading. However, the question is, as non-experts, is there the possibility of restoring PCDH19 mosaicism? And can this be achieved? Is it possible to, for instance, induce expression in the hippocampal cortex? And is this the same strategy? Can this give us the same outcome? The answer probably is no, because we're looking at two different strategies. I don't know whether they are complementary, and they have to be tested independently. And we know that there is initial evidence with DMRI studies, as illustrated by Dr. Canale, and this is our hope. We hope that if we reactivate the protein and recover its functionality, even in the presence of anatomical structures that have been formed in a pathological and non-physiological fashion, maybe restoring, let's say, this protein, if it is still able to generate gene expression, if it maybe some symptoms can be attenuated and some outcomes can change. And Joseph showed this in the last slide. It's possible maybe to intervene in a already formed brain. So we would like to invite you to act along these different paths. we're not in this area of research, but we appreciate what you do, and we're very fortunate to be able to read your papers and reports. And this is just my concluding slide. I would like to thank you all. I would like to thank today's speakers. 2 years ago was my first time at one of these conferences, and together with Francesca, I had the honor of selecting abstracts, which will be featured in tomorrow's session. A lot has been achieved in a very limited amount of time, and I would like to, and we're all tired, I know. I would like to end by thanking the scientific community that is here for the precious contribution. Especially, I would like to thank Joseph, Maria, and Jack. You saw Joseph a short while ago. I'd like to thank them for all the time they spend interacting with us. And this is not something we take for granted. We always say this with Francesca. And we know that at times we ask questions that a researcher will find naive. And they might say, oh no, can't do. But they really are amazing. Maybe we're talking about more than 100 emails, maybe 200. We've exchanged hundreds of emails, and they've always replied in between conferences. And this is not something we take for granted. And because of this, we thank you publicly. Thanks. And we thank all of you.